All right, let's welcome back. Let's move on to chapter 6. It's an interesting chapter from the standpoint we know that these bacteria, primary goal in life, as I assume, is not to make you sick. It's just to grow in a niche. If that happens to be you, then, well, so be it. So, our job is to understand how they do this. And unfortunately, or fortunately, however you like to look at it, we got to look at the biochemistry of the microorganism. And instead of saying biochemistry, we'll just call it nutrition. And what does that entail? So, our objectives is that we got to look at the essential nutrients that bacteria need. And we're going to talk about macro and micronutrients in terms of the amounts that they use. We'll talk about terms that describe the organism's sources. Now, in lingo of a microbiologist, we say, well, this sugar is this carbon source. So let's say sucrose is the carbon source that this one uses, and blah, blah, blah. You can actually, based on their sugar utilization, help identify them. That's one of the tools. So understanding what their biochemical traits are important in identifying them, in addition to how we manipulate them and that sort of thing. And that, of course, how it breaks down the carbon or the sugar is actually how it derives its energy. So we need to know that. What is a saprobe, a parasite, and explain why these terms can be an oversimplification, and that's always the case, seems like. And we're going to compare diffusion and osmosis, and my gosh, I would imagine you've been through this so many times. I have a slightly different take on it, and so it might be of interest to you. Isotonic, hypo, and hyper. Of course, we're looking at the effects of salt in the environment, typically, is what it boils down to. Um, I named two types of passive transport. So hopefully you already are aware of this. So this is 6.1, and let's get going here. So an essential nutrients, any substance that must be provided to an organism. Macronutrients are required in a relatively large quantity. That's why we call it macronutrients. It's referring to the quantity. And of course, these would be high utilization type of things that they put in their cell walls and various things, part of the cell structure, the infrastructure, and then of course they use it for metabolism, which is just to extract chemical bond energy when it needs it, right? So in these, typically a carbon, hydrogen, and oxygen containing, sort of like C6H12O6, carbohydrate, things like that. Micronutrients present in much smaller amounts and are involved in enzyme function and maintenance of protein structure. So there's some things that we can string all we want, the amino acids, together, but they're not going to have the characteristics and properties that are required to do whatever it is. We need that additional bonding strength or reactivity or whatever it is. And we refer to these micronutrients sometimes as trace elements. And so it's something to think about. And trace elements are important. Manganese, zinc, nickel, we have the same sort of requirement for the same reason. Certain things that our bodies need to do the chemistry that cannot be achieved with just the amino acids. So we have to have these cofactors that go with enzymes and various things to make life happen. So inorganic nutrients, an atom or a simple molecule that contains a combination of atoms other than carbon and hydrogen. So they're found in Earth's crust, in metals and their salts, magnesium sulfate, ferric nitrate, sodium phosphate. So a lot of these are making the chemical structure more stable by satisfying all the bonds, the octet rules and things, by adding the sulfates and the nitrates and the phosphates. In that way they kind of are more manageable in some ways in terms of stability and that sort of thing. Gases: oxygen, carbon dioxide, and water. And those are the two important aspects, of course, of inorganic nutrients. Is of course water, H2O, or oxygen, hydrogen, hydroxyl group, and hydrogen, which makes up the pH. In a long way, I think we've already gone over that. Organic nutrients contain carbon and hydrogen atoms that are usually products of living things. Well, yeah. A simple organic molecules such as methane, or if you're from Great Britain, methane. Large polymers, carbohydrates, lipids, proteins, and nucleic acids. So they're long chains of polymers, repeating structures, and that's a good thing. That's how we save, store, build structures, and that sort of thing. So microbial cytoplasm, the gooey center of a bacteria cell, is 70% water. It has protein. We sprinkle in a little bit of organic compounds, which is, just 97% of its dry weight. Carbon, hydrogen, oxygen, nitrogen, potassium, and sulfur, or CHOMPS, you might want to say, 96% of that. Most chemical elements available to the cell as compounds and not as pure elements. And those are the salts and things that we were just talking about. Only a few types of nutrients are needed to synthesize over 5,000 different compounds. Isn't that amazing? So that's the purpose. And so here's the chemical composition of proteins, the percent, weight, nucleic acids, RNA, DNA, and then you look at carbohydrates, lipids, and whatevers. And then you look at carbon, oxygen, nitrogen. Those are the big three out of that whole list of elements. Hydrogen, of course, that satisfies the octet rule, saturation, things like that. Phosphorus, sulfurs, a lot of times there's cross-linking, like the structure of an antibody is cross-linked with sulfur that gives it that stronger Y-looking shape that we've all come to love. We'll talk about that in chapters 12 and 13. Sodium, calcium, magnesium, and all of these different types down here. Trace metals make up about 0.3%, so that's really small amount. Iron, I'll just take a moment why it's important to study these things. Iron is one of the most important aspects. This is where a lot of the organisms that get into our system, into our blood, and trying to set up a homestead. Iron actually becomes a point of contention because we need the iron too. We use the iron for lots of reasons. One that you may not be aware of, but iron is required as part of an enzyme to add the amino acid onto a transfer RNA, and we call that charging of transfer RNA. So it's all about iron. The battle, when it comes down to the absolute push and shove, we have certain affinities for the iron, and we pull out that iron in our nutrients and various things as we eat and do things, and we get it in the blood, and we hold on to that iron, and we need it for, of course, heme, for binding oxygen, and also very, very small amounts of this iron that has to be kind of free around in order for those enzymes, when they're made, that charge transfer RNAs. And if we don't have that irons available, then that's a problem. We can't build amino acids, and so we have to have that trace amount of this iron, and it's right on the edge of being traced. But anyhow, iron's there, and so it now becomes a battleground. And there's several researchers in large areas of pathogenic bacteria that do the research about those. Really very, very interesting. If you want to tear into that, you can just do iron and pathogenicity search, and you will find a plethora of papers related to that. So it's pretty much, if you look in our own history as human beings, way back, I don't know how many years ago, it used to be you could see paths to like salt mines, and you have to have salt in order to survive. And so part of the trade and the way the organization of humankind was kind of centered around getting key essential nutrients and various things, and salt was one of them. So iron was important. So I may bring up a paper too about the iron aspect, because it really is very, very important. There's some really major pathogens that it boils down to iron chelation, or out competing our own ability to sequester iron, which is really interesting. Okay, enough about that. Heterotrophs, organisms. We've already been through this several times, but I'll do it again because it's important. I mean, a heterotroph, okay, an organism that must obtain its carbon from an organic form. Autotroph uses inorganic CO2 as its carbon source. A phototroph is one that photosynthesizes, and a chemotroph gets its energy from chemical compounds, and there's examples of all of these. So let's go and look at this. Autotrophs. A photoautotroph, obviously we add the word photo, then we know sunlight, and it's algae, plants, cyanobacteria. Chemoautotroph, organic compounds. So how does that relate to the autotroph? Is an organism that uses inorganic CO2. So carbon sources, you can see, even though it utilizes sunlight in these the carbon source is CO2, and so that's incorporating that carbon, and releases oxygen in addition to the cracking of the water when we do the photosynthesis part. Heterotrophs, photoheterotrophs, a purple-green photosynthetic bacteria. So you can see photoautotroph, and then the difference between photoheterotroph is its carbon source. So really what we're delineating is this further grouping of auto. We move down to photo, or heterotrophs, we move to photoheterotroph. It means it utilizes the energy source of sunlight, and its carbon source is organic. So I just wanted to point out that's the sort of the determining factor. So if you look at this chart from your book, or you can see this, it's important. I may post this one as a handout on Blackboard for you, just so that you'll have it. So they're interesting examples, but I just wanted to make that distinction. So when you see the word being built off of heterotrophic chemo, and then saprobe and parasite, saprobe lives off of dead things, and a parasite is one wins, one loses, and the one that loses is usually the host, right? The one winning is the one eating you. And so anyhow, there's some of the delineations. So photoautotrophs are photosynthetic, and they produce organic molecules using CO2. So you can see that chemoautotrophs are chemoorganic autotrophs, use organic compounds for energy and inorganic compounds as a carbon source. So there's sort of a very important litho. Autotrophs rely totally on inorganic. When you think of litho, think of stone or rock. So it's just, if you have that thought, so it's sort of the Latin meaning of that. Inorganic materials require neither sunlight nor organic nutrients. So those pretty tough. Chemoheterotrophs derive both carbon and energy from organic compounds, and they process these molecules through cellular respiration or fermentation. So you can go back to that table and just refer. Sometimes these you can get a little bit lost. Saprobes, free living, that feed on organic debris from dead organisms, and just saprobe. They're feeding off of dead things. It decomposes of plant litter, animal matter, and dead. Well, we'd be up to our elbows in dead things if we didn't have saprobes. And so they recycle organic materials. We've got to have that. Parasites derive nutrients from cells or tissue of living hosts. Pathogens cause damage to tissues and even death. So we refer to these as parasites, or ones that bother us a lot. No, you don't quote me on that one. Just parasite. Pathogens are a little bit more of a delineation where they're causing damage. Because they wanted to set up house in your tissues, there's waste products and things that damage, or they utilize your tissues as a carbon source or whatever. Parasites range from viruses to helminths, so we can use viruses and worms. Ectoparasites live on the body. Ecto, I mean, like ectoderm is the outer protection of an animal. Endoparasites live in the organs and tissues, those are inside. And intracellular parasites live within the cells itself. So these I do want you to know, so you can highlight these. It's important to add that to your repertoire of understanding. So let me go back. So parasites, obligate parasites unable to grow outside the living host. Leprosy, bacillus, and syphilis spirochetes are examples of obligate intracellular type parasites. Believe it or not, leprosy, there's still leper colonies, and don't be surprised. Uh, you think that was eradicated, but it hasn't, and it's still around. And syphilis, well, yeah, unfortunately, some of the new social medias have spawned folks and being able to get syphilis a lot easier than they used to. That's not good. So some of the essential nutrients. Carbon, most common organic molecule that satisfies protein. Carbon's a building block because it needs four more somethings to fit in to satisfy the octet rule, and it makes it a really nice Lego or a building block in order to build bigger things. Hydrogen's a major component of inorganic and organic molecules: water, carbon, hydrogen, calcium, natural occurring gases, hydrogen sulfide, methane, hydrogen, atmospheric hydrogen, those sorts of things. Um, so calcium hydride, dihydride, I guess that is. These gases are both used and produced by microbes, of course. Hydrogen helps maintain their pH because a breakdown of water, OH and H, the percent hydrogen really dictates how the pH, as you recall from our previous discussions. 100% hydrogen is zero, pH is zero, and then zero amount of hydrogen is a pH of 14, which means that we have 100% of hydroxide. So you can see the inverse relationship there. As a review, so useful for forming hydrogen bonds between molecules, also serves as a source of free energy in respiration. Oxygen is a major component of oxygen or of organic compounds, such as carbohydrates, lipids, nucleic acids, and proteins, and its structural role and enzymatic functions of cells. Oxygen is likewise a common component of inorganic salts, as sulfates, phosphates. We have these OH groups or phosphorus groups, all sorts of things that retain the oxygen part. Free gaseous or metabolic or atmospheric oxygens, O2, that makes up about 20% of the atmosphere, which, by the way, if you give microbes millions and millions and millions of years, that's where we got the oxygen in the atmosphere. I know because I was there when it was very low. Just kidding. Nitrogen, a major reservoir of nitrogen gas, which makes up 79% of the Earth's atmosphere. This element is indispensable for the structure of proteins, DNA, RNA, ATP, which all are important, as we know. Hopefully you recognize all of those. Such compounds are primarily nitrogen source for heterotrophs, but also useful. So not only do we have to have carbon sources, we have to have nitrogen sources, because this is indispensable for the structure of proteins, DNA. So you see the requirements echo exactly their needs in order to build structural or do enzymatic activities and various things like that. And so the nitrogen source is useful. They must be degraded into their basic building blocks so they can be incorporated into other things. Some bacteria and algae utilize inorganic nitrogenous nutrients, so NO3 and the like. Ammonia. A small number of bacteria and archaea can transform atmospheric nitrogen into compounds usable by other organisms that process nitrogen fixation. Azotobacter vinelandii is an example of a microorganism that nitrogen fixes, and it can literally pull atmospheric nitrogen and then incorporate it. In fact, there are certain bacteria that is absolutely associated with the roots, is required with the roots of some plants. So they sat down with plants and negotiated a deal. Well, actually, I don't know about that part, but there's certain bacteria that will nodulate the roots, and they will do the nitrogen fixation for the plants. So you essentially have self-fertilizing plants because of the bacteria being associated with the roots. And this is really, really cool. So a lot of times farmers will rotate certain plants. It's like soybeans, and they may have corn many times, and then they can rotate and plant soybean, which becomes nodulated with bacteria that do the nitrification, and that helps to nitrify that plant and the soil. And then you can go back and plant corn, and there you go. You've already fertilized, and you don't have runoff, and it's a cheaper way to do business, and it works. Phosphate, the main inorganic source of phosphorus, is phosphate. Is derived from phosphoric acid, H3PO4, and is found in rocks and oceanic mineral deposits. Phosphate is a key component of nucleic acids, of course we know that. And when we think about linking various phosphate groups with double bonded oxygens as a sort of a group of three, like adenosine triphosphates, a chemical bond energy, 32 kilocalories per mole to break one of those bonds, serves as a cellular energy transfer. So it's chemical bond energy stored in here, and phosphate helps accentuate that particular chemical bond energy. Other phosphate-containing compounds are phospholipids, really abundant. I mean, you think about every single cell is phospholipid bilayers. And then the coenzymes such as nicotinamide diamine, NAD. Sulfur is widely distributed throughout the environment. Rocks and sediments such as gypsum contain sulfides and sulfates, hydrogen sulfide gas, various things. Vitamins B12, B1, these contain sulfur, and some amino acids, methionine and cysteine. The latter helps determine the shape and structure of proteins by forming unique linkage called disulfide bonds. So anytime we need that, so you can imagine that in the antibody example I've already mentioned, is that we use sulfur. Well, you would imagine all you'd have to do is have precisely positioned amino acids. So on one of the aspect of that antibody would have a placement of a cysteine, and then another one, so you can create that disulfide bond, and that's in fact what happens. Other important nutrients are potassium, essential for protein synthesis and membrane function, of course. Sodium, important for certain types of cell transport. Both the potassium and sodium, I think you remember from anatomy, the key pieces of these things for facilitating electrical functions of muscle contractions and blah blah blah. Calcium, same thing, stabilizes cell walls and endospores of bacteria. So they're important, but maybe for different reasons in bacteria. Magnesium, component of chlorophyll and a stabilizer of membranes and ribosomes. And magnesium is again widely used. Iron, a component of the cytochrome proteins of cellular respiration. Yet another important aspect of iron is for cellular respiration. Zinc, essential for a regulatory element for eukaryotic genetics. The zinc fingers, they're called, and various things are important for stabilizing genetic structures and doing enzymatic manipulations like DNA maintenance and that sort of thing. How microbes eat. The transport of necessary, just literally moving across the membrane, occurs even in organisms with cell walls. So they have mechanisms that pierce or allow only certain things to get in. The driving force of transport is atomic and molecular movement. Diffusion is a phenomenon of molecular movement in which atoms and molecules move in a gradient from an area of higher concentration to a lower area of concentration. and that's diffusing. In other words, all I have to do is take some maybe methylene blue, drop it into a glass of water, and you'll see it diffuse. And that's diffusion, and driven by Brownian movement, which is the random collisions between atoms because of being at room temperature. There's ambient heat, and it's moving the process. So osmosis is the diffusion of water selectively or differentially through a membrane. And that's the key with osmosis, and it's really interesting. Has passageways that allow free diffusion of water but block certain other dissolved molecules. When the membrane is placed between solutions of differing concentrations of solute, and solute cannot pass through the membrane, water will diffuse. So what's really driving the whole process of osmosis is the selective movement of water to accommodate differences in osmotic pressure, which is just the effects of water to gravity, and that's what drives that diffusion of the water passing through a membrane. Things that can't pass through it, of course, are taking up valuable space, and so the water has to compensate by making up for that space. That is osmosis from your professor's point of view. This will continue until the concentration of water is equalized. So there you go. Osmotic pressure is the effects of gravity and on the water, and it will equalize on both sides of the membrane at a given time. So you can see there's more solute inside of this membrane and then outside, and so what's going to happen is the water is going to try to equalize in terms of the pressure, and when it does, it's going to be adding more water, and you can see it driving up the glass tube there because these red things are displacing that water, and so that exact amount of area that it's taking up is compensated by molecules of water, and hence you see more in there. And again, that's driven by the forces of gravity and water diffusing. That's all it is. And then you can see the little zoom in there to help visualize that, and that works. And then the cell's response to osmosis, iso is the same. Hypo, like a hypodermic needle, and hyperdermic. So hypo is below, hyper is above. And so if you remember, cells with a cell wall, cells without a cell wall, is just the same process, but has a little bit more of a meaning, I guess, in some ways. But hypotonic is that water is moving into the system. So for whatever reason, is trying to compensate by moving water into the system to get it to look more like the exterior. And the same with the others, but you see it more profound with cells without a cell wall. And then hypertonic is that we're trying to add water to the environment to equalize that pressure, and a lot of times it shrivels up. And I understand this one. I was bitten by a cat when I was working at a vet clinic, and I was in for rabies observation. I've already alluded to that, but the story thickens because when I was bitten, it went through my nail in my hand, and I went into the hospital because I was within hours. I had red streaks up my arm from an infection, and so they had to pull the nail, and so it was during a nursing station, I guess, was going through a nurse change, and the doctor thought I had gotten a numbing agent from my thumb, and I didn't. So he yanks the nail, and of course that felt really good. He wouldn't listen to me. I said, "Doc, he didn't listen. He just pulled it." So anyhow, when I started to faint because it was painful or whatever the reason, they gave me an IV, and they forgot to put the salt solution in it. I guess it was Ringer's, and you had to supplement 0.95% salt, because if you don't, then the cells will crenate. And again, that just added to my fun for that day. That was one long day, let me tell you. But anyhow, lest I digress, the idea was that the cells crenated because we had a situation where things were not good. Okay, passive and active transport. Passive doesn't require energy, active does. So passive is simple diffusion or facilitated diffusion. Now I argue a little bit, and I had students of over time. Facilitated diffusion. Well, didn't the cell have to create the protein, and that took energy to do that? And so in a way it invested that energy in proteins that move, and yes. But what they're referring to in this is, in order to have that movement, we have to pay to play. In other words, it requires a quarter. And these don't require anything. So in other words, the passive is because no matter if that protein had to have energy to make it and all that before, we're actually talking about the transport process, facilitated diffusion is facilitated by a protein that moves things in. And it does that by, when it binds, it changes its shape, so it pushes that substrate through. So that's fine, and that's the facility. It's sort of like a turnstile and that sort of thing. The idea, though, is it's molecule specific, and so it only is bringing in stuff that it wants or whatever it was designed to do. Active is carrier mediated, but you have to pay to play. In other words, in order for it to move in, we have to drive it by the chemical bond energy from ATP. And so there's kind of the distinction, and I'm sure you've been through this many times. So it's usually against the diffusion gradient, so we're going uphill, so we have to force the issue, and we do it at a faster rate than normal. The presence of specific membrane proteins like permeases and pumps actually do the transportation, and then of course in order for to facilitate the shape change that drives that pump or whatever it is, we energize that shape change by chemical bond energy of ATP. So as we break that bond, that energy is released into the molecule such that other types of relationships and bonds change as a result of that chemical bond energy and change its shape and facilitate some movement. And then when that's done, it goes back. It's sort of like a hinge, one way and the other. And there you go. I hope that makes sense. Eating and drinking. Endocytosis. Cell encloses the substance. Now we've talked about endocytosis when we talked about viruses and how they trick a cell into thinking that it's something it wants, and it pushes the doorbell and it opens the door, and there you go. Spontaneously forms a vacuole and engulfs the substance. So that's endocytosis. Phagocytosis. So endo means within the cell. So it's a process by which the cell brings things in. This is a process by which cells eat. And that phago just means eat in Latin, so accomplished by amoebas and white blood cells, ingest whole cells or large solid matter. Pinocytosis is the process of a cell drinking. Now you think of it drinking a pint of beer, so now pint should be in your mind in terms of the Latin term pinocytosis, so ingest liquids it's drinking, such as oils or molecules in solution. And there you go. So believe it or not, I know you've had so much fun joining me with 6.1. I will stop here, and that way you can use the facilities or whatever you need to do, and we'll take a break, and then I'll come back with part 2 talking about 5 terms used to express the temperature-related growth capabilities, microorganism functions in differing oxygen conditions, and there you go. So not so bad. This is pretty straightforward stuff. A lot of this you've already had, the diffusion and active and passive and all that stuff. So I hope you're well, stay well, and I'll speak with you next time.